Okay, hello everybody. So I will be starting my class today. I will start with a YouTube video and then I will go with my lecture. Today is the last class possibly, that glycogenolysis in my portion. So uh, let's start.
Hi, in this video, I'll be talking about glycogenolysis. So if we break down the term, it's glycogen and lysis. So basically it means breaking down of glycogen. So simply, glycogenolysis is the process by which big glucose polymer, or you can imagine them as glycogen, is broken down into single glucose residues. But before understanding glycogenolysis, we should ask ourselves, where does it take place, when does it take place, and how does it take place? So let's answer these questions in this video to understand the concept behind glycogenolysis. And in this video, I would also give a real life example where glycogenolysis takes place, and it is essential for your body's physiology. So stay tuned till the end of the video. Now, the site for glycogenolysis is muscle and liver. So that answers our question, where does it take place? But why does it take place in muscle and liver? Simply because all the enzymes, such as glycogen phosphorylase, debranching enzyme, and phosphoglucomutase, these enzymes, which are essential for glycogenolysis, is present in the liver and the muscle. And second of all, the substrate. That means glycogen, which is broken down into glucose, is actually stored in muscle and liver as a reserve for glucose. So that is why this is the primary site for glycogenolysis. Now then ask that, when does it take place? One of the situation when glycogenolysis is important is starved state. So in starved state, what happens is, like, body is uh, deprived of any kind of external nutrition intake. So bodies has to, like, break down its own glycogen reserve in order to produce energy. So simply,
in the south of Odisha, on the border of Andhra Pradesh,
the liver glycogen and the muscle glycogen would be broken down in order to produce glucose, and that could be used in the body to produce energy. And especially brain need glucose as a fuel, right? So let's talk about glycogen a bit. So glycogen is like a globule. So it has one particular protein known as glycogenin, and that works like a nucleation factor around which the glycogen polymer, which is basically a chain of glucose, exists. And this glycogen is a branched polymer. Now let's just simplify that structure to this way. So glycogenin is linking all the branched polymers together and forming like a globular structure. So the glycogen has two major kind of linkage. One is alpha 1,4 glycosidic linkage, that is the straight chain, and the branch chain has alpha 1,6 glycosidic linkage. Why this is important, I'll talk in a bit. So the key enzyme for glycogenolysis is glycogen phosphorylase. So glycogen phosphorylase basically sits on the alpha 1,4 glycosidic bonds, and by an enzymatic reaction it breaks the bond, and that is why that is how it free away the glucose residue. And also there is a phosphate group in the glycogen phosphorylase which basically phosphorylate the glucose and forms a glucose 1 phosphate residue. That's the product. Now it proceeds through the branched end till fourth amino acid before the fourth glycogen residue before the branch point. So let's talk about the glycogen phosphorylase in a bit. So glycogen phosphorylase has a pyridoxyl group, or PLP, which is basically derived from vitamin B6 at the catalytic site, and it has two catalytic sites, and each of these catalytic sites has PLP, and PLP has a very important role in the enzymatic reaction mechanism. And it forms a CIF base with the basic amino acid lysine 680, and in a moment we would understand why it is important. So glycogen phosphorylase breaks alpha 1,4 glycosidic linkage, right? So in in a couple of minutes we would understand that what is the mechanism behind that. So here is glycogen phosphorylase, and here is the active site of glycogen phosphorylase. So what you can see is that the phosphate, the oxygen from the alpha 1,4 glycosidic linkage is basically getting protonated, and further it gets a nucleophilic attack from the phosphate group itself, which would make the glucose into a glucose 1 phosphate and leave the glycogen with one less glucose residue. And this is how progressively the glycogen polymer would be degraded into glucose 1 phosphate residues. Now, whenever the glycogen phosphorylase gets to the end of a branch point, at least 4 residues away from the branch point, it halts. Now then glycogen phosphorylase gets replaced by a debranching enzyme. Now debranching enzymes are special because debranching enzyme can break the alpha 1,6 glycosidic linkage, which glycogen phosphorylase can't break. So debranching enzyme would break the alpha 1,6 glycosidic linkage and make it a linear chain of glucose right now. And once it is a linear chain of glucose, our glycogen phosphorylase can again attach and start breaking down the chain again. So this is how glycogen phosphorylase is one of the rate limiting enzyme in glycogenolysis, and we know how. Now once the glucose residues are broken down and in format of glucose 1 phosphate, now let's see what happens to this glucose 1 phosphate. So glucose 1 phosphate, with the action of phosphoglucomutase, is getting converted into glucose 6 phosphate. Now phosphoglucomutase has a phosphate group. Now this phosphoglucomutase is getting a nucleophilic attack from the oxygen which is attached to the carbon number 6 in the glucose 1 phosphate, and as a result what has happened, it is forming a transient intermediate known as glucose 1,6 bisphosphate and forming a serine residue instead of a phosphoserine residue in the phosphoglucomutase enzyme catalytic site. Now a further reaction, which is another nucleophilic attack to the carbon residue 1, converts this whole thing to glucose 6 phosphate. So that is how, by the enzymatic action of phosphoglucomutase, glucose 1 phosphate is getting converted into glucose 6 phosphate. Now glucose 6 phosphate is a very important intermediate because glucose 6 phosphate can either enter the glycolysis, and in the glycolytic pathway it can form the pyruvate, or it can also enter the pentose phosphate pathway where it can produce NADPH and ultimately give rise to ribose 5 phosphate, which could be used for nucleotide biosynthesis and all. So how the glucose 6 phosphate would be utilized, it's the matter of body's metabolic demand. For example, if the body requires energy, it would quickly use the glucose 6 phosphate into pyruvate and produce energy that would provide body instant energy. But if the body needs anabolic responses such as fatty acid biosynthesis, cholesterol biosynthesis, etc., then body would produce more NADPH, or body needs a lot of transcriptional activity or a lot of gene expression to be happened, then it would choose the pentose phosphate pathway to channel the glucose 6 phosphate into that pathway. Now it entirely depends where the glucose 6 phosphate would be channeled, in pentose phosphate pathway or in glycolysis, and it all depends upon the external demand. So let me give you a current example. So let's say you are standing on a dog's tail and the dog is like mad over you and trying to chase you, and you are like hell scared. So definitely your muscles need to move, and immediately your muscles need a hell lot of glucose. But after a while your muscle glucose is depleted, but still you need to run to avoid the dog bite, and that is why you need to break down glycogen in your muscle to produce glucose, and that could power up your muscle, right? So one hormone, which is epinephrine or adrenaline, it's regulating the glycogen breakdown upon this kind of flight or fight situation. So adrenaline binds to adrenergic receptors on the muscle cells, and in a G-coupled signaling mechanism takes place, which ultimately leads to cyclic AMP generation, and cyclic AMP dependent kinase, or pKa, is activated. The pKa catalytic subunit actually activates phosphorylase kinase, and phosphorylase kinase is an important regulator enzyme. So whom does it regulate? Phosphorylate kinase actually phosphorylates the glycogen phosphorylase. Now we have already learned that glycogen phosphorylase is so important to break down the alpha 1,4 glycosidic linkage and thereby breaking down glycogen into glucose. So thereby the hormonal control to an immediate flight or fight response would increase the phosphorylase activity, which can eventually break glycogen into glucose and give the muscle instant source for glucose to channel it into glycolysis pathway and Krebs cycle to get the energy, and that would pump your muscle up for running and save your life, right? So that is how the hormonal control is important over metabolic pathways in order to save our body's physiology. So I hope you enjoyed this video. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.
So this is from an animated biology with Orpon. So from this YouTube, this is nicely explained, a very nicely explained part this particular video. And so let's get back to my topic. And so while we after this, let me go back to my topic, and that is glycogenolysis. So as it says, the degradation of stored glycogen in liver and muscle constitutes to glycogenolysis. So the pathways for synthesis and degradation of glycogen are not reversible. An independent set of enzymes present in the cytosol carry on out glycogenolysis. So you have seen glycogenesis, that is formation of glycogen from glucose, and breakdown of glycogen into glucose. They are reversible pathways, but this is not reversible. There are different enzymes which do this. So glycogen is degraded by breaking alpha 1,4 and alpha 1,2,6 glycosidic bonds. And glycogen is so there are two types of linkages as shown in the video. One is all these bonds which of these glucose are joined together by alpha 1,2,4 glycosidic linkage, and the branch point is alpha 1,2,6 bond. So the enzyme is glycogen phosphorylase. The enzyme which is used is glycogen phosphorylase, and this breaks down these particular alpha 1,2,4 block as 4 units. So when around 4 glucose units are left from the branch point, that is where alpha 1,2,6 bonds are present. For this particular compound is known as limit dextrin. Then there is an enzyme which has got transferase activity, that is the debranching enzyme. It has got alpha 1,2,6 glucosidic activity. So what it does, it removes these 4 groups. This is transferred here and it comes over here, and then again it is transferred here, and then alpha, the further action of this particular thing and free glucose, this free glucose which is present over here, as you can see, this is a free glucose which is attached over here, to which is attached, say for 3 glucose molecules. So this is first transferred at its back, so this remains nakedly present over here, and in the end you find that these 3 are there, and then this free glucose is released by alpha 1 to 6 glucosidic activity. So this is removed out, this free glucose is removed out, and then further activation of glucose phosphorylase, it breaks it into glucose 1 phosphate, small units of glucose 1 phosphate, and now glucose 1 phosphate in the liver is converted by phosphoglucomutase enzyme into glucose 6 phosphate, and then which it is broken down into glucose. So first is the action of glycogen phosphorylase, and that is that breaks the 1 to 4 linkages. It breaks till 4 units of glucoses are attached over here. Then the next thing which happens is the presence of debranching enzyme, the transferase, which transfers the branched this branched unit over here. It is transferred to this end, and it becomes straight, and 1 to 6 glucosidase activity, it is cut, and so free glucose is released, and then phosphorylase keeps on cleavage, alpha 1 to 4 cleavage starts to take place. So when is glycogenolysis promoted? When the body requires a lot of amount of glucose. So when you are running, or when you are in desperate need of energy or that glucose, then glycogen is broken down. So this is how it is also regulated. So these are the enzymes, and these are all the steps. So there is an allosteric regulation of glycogen metabolism. So glycogen phosphorylase is the enzyme which breaks down glycogen, and glycogen which makes glycogen is glycogen synthase. So when there is lot of glucose 6 phosphate, it promotes the formation of glycogen synthase, because lot of glucose is 6 phosphate is present. Glycogen synthase is activated and allosterically activated, and it makes more glycogen. Lot of glycogen, this activates glycogen phosphorylase. Glycogen phosphorylase, when there is a lot of glucose 6 phosphate here, it negatively inhibits the activity of glycogen phosphorylase. That is an allosteric inhibition. And ATP, lot of ATP present, it stops, because you do a lot of ATP present, you don't need glucose, then you have ATP. Then lot of liver glucose, it inhibits negatively. Lot of cyclic AMP calcium, it positively stimulates glycogen phosphorylase. Lot of AMP, because AMP means it needs more ATP. So this is promoted. Glycogen phosphorylase promotes the breakdown of glycogen into glucose. So this glucose can be, glycolysis can take place, and ATP can be released. So this is how actually the whole system of glycogenolysis takes place. This was a very short topic which is remaining, and with this I stop sharing my screen. Let's see if anybody has got any questions. Please ask your questions, and next day, if there is time, I will tell you, I will be taking special classes on other topics of biochemistry which you may be interested. For this I have to give notice. So my portion has been finished in biochemistry. The portion which has not only my portion, I have finished Shashwati madam's portion also, because she is on maternity leave. So she her portion also I have taken up. I have I have covered the carbohydrate metabolism, all the topics of carbohydrate metabolism, all the topics of lipid metabolism. Now who is taking protein metabolism? Can anybody tell me? Anybody is taking protein metabolism? কে প্রোটিন মেটাবলিজমটা কেউ নিচ্ছে তোমাদের? হ্যালো,
স্যার মনে হয় দ্বারকা ম্যাম নিচ্ছে। মনে হয় আমি এখন অব্দি দেখিনি, মনে হয় স্যার আমি সিলেবাস ওটা দেখিনি, মানে কার কোনটা অ্যালটেড আছে, পিডি এখন খুলতে পারছি না তাই
কেন
জন্য।
আমার নেই, ওটা প্রোটিন মেটাবলিজম জারকা ম্যাডাম নিচ্ছে, যদি না নেয় আমাকে জানিয়ে দিও। তোমাদের কি অন্য কোনো টপিক আছে আমি পড়িয়ে দিলে ভালো হয়?
স্যার অ্যানাটমি পোরশনটা পড়ি না, আর কি আছে স্যার, রিভিশনই করিয়ে দিন আপনি, মানে পুরো বায়োকেমিস্ট্রি ধরুন, ওই কোয়েশ্চেন কিরকম 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 কোয়েশ্চেন আসবে
বায়োকেমিস্ট্রি
ওরকম সব।
পড়াবো বলছো?
হ্যাঁ হ্যাঁ,
অন্য টপিকগুলো পড়াবো?
অন্য টপিক গুলো থেকে স্যার কোয়েশ্চেন কিরকম আসবে ওগুলো
কোয়েশ্চেন কিরকম
শর্ট
আসবে?
রিভিশন করিয়ে দিন। হ্যাঁ, শর্ট রিভিশন টাইপ করিয়ে দিন, কারণ যে এক্সাম তো এমনি কাছে চলে এসেছে তো তাই জন্য।
আচ্ছা, এই ছবিতে তোমাকে খুব রোগা লাগছে।
হয়ে গেছে, এখন কিছু করার নেই।
তুমি কি এখন রোগা হয়ে গেছো?
হ্যাঁ
ও বাহ, বেশ ভালো লাগছে, সুন্দরী লাগছে, বাহ খুব ভালো। কিপ ইট আপ, হেলদি থাকবে, যত স্লিম ট্রিম থাকবে
হ্যাঁ
তত ভালো।
হ্যাঁ স্যার।
আচ্ছা, তাহলে বায়োকেমিস্ট্রিটা তাহলে একটু কি কি কোয়েশ্চেন আসছে, কি কি টাইপ, কি কি পড়বে, এই টাইপ এই জিনিসগুলো একটু বলে দিলে তোমাদের ভালো হবে, তাই তো? কি কি টপিক
হ্যাঁ স্যার।
পড়বে? ঠিক আছে, তাহলে নেক্সট দিন ক্লাসেতে আমি ওইটাই আমরা ডিসকাস করব, কি কি পড়তে হবে ভালো করে বায়োকেমিস্ট্রিটা, ওই জিনিসগুলো যেন ডেফিনেটলি পড়ে যেও তোমরা।
ঠিক আছে স্যার।
বাকিগুলো সবই পড়তে হবে, কিন্তু কোন কোন জিনিসগুলো আমার মনে হয় আসলে মুশকিলটা হয়ে গেছে, আগের বছর বন্ধ ছিল না, কোয়েশ্চেন পেপার কি বসিয়েছে ভগবান জানে। আগের বছরে কোয়েশ্চেনটা তো তোমাদের তো কেউ পরীক্ষা তো আগের বারে দেয়নি, হয়নি তো পরীক্ষা। তো যে
হ্যাঁ ওটা
একটা
অনলাইনে হয়েছে
পেপার
তো
সেট হয়েছে,
অনলাইন।
একটা পেপার সেট হয়েছে। এবারে
আচ্ছা
ব্যাপারটা কি
আচ্ছা
হয়েছে, যাকে বায়োকেমিস্ট্রি এবারে পেপার সেট করতে দিয়েছে তাকে আগের বছরের কোয়েশ্চেন পেপারটাও দিয়ে দিয়েছে। হ্যাঁ, এবারে সে সেই পেপারটা পাবে, আমি তো পাইনি সেটা। আর সেটা এখন সেটা দেখে সে কোনটা দিয়েছে না দিয়েছে, আমাদের আমার আমার এবারে গেস ওয়ার্কটা, যদি আগের বছরের পেপারটা পেতাম তাহলে ওইগুলোকে আমি বলতাম যে ওইগুলো একটু কম করে পড়ে যেতে, কারণ ওগুলো তো
আচ্ছা
আগের বারে
আচ্ছা।
এসছে, সাধারণত রিপিট করবে না, অন্য
হ্যাঁ
অন্য
হ্যাঁ।
জিনিসগুলো
হুম হুম।
পড়তাম। হ্যাঁ, কিন্তু এবারে আগের বারে এসছে, ওটা তো ডিক্লেয়ার্ড হয়নি আগের বারে কোয়েশ্চেন পেপারটা, কিন্তু সেই
হ্যাঁ হ্যাঁ।
কোয়েশ্চেন পেপারটা যখন পেপার সেটারকে দেবে, আগের বারে কোয়েশ্চেন পেপারটা পেপার সেটারকে দেবে, তো পেপার সেটার দেখবে যে আগের বছরে এই এই কোয়েশ্চেন এসছে, কিন্তু অ্যাকচুয়ালি আসেনি কোয়েশ্চেনটা, আমরা কেউ জানি না সেটা। হ্যাঁ, সো মুশকিলটা হয়ে গেছে, সো উই ডোন্ট হ্যাভ, মানে আমাদের একটা ব্লাইন্ড কার্ডস খেলতে হবে, উই হ্যাভ টু প্লে ব্লাইন্ড কার্ডস।
হ্যাঁ বাট তাও সিলেবাসের মধ্যে থেকে তো দেবে, কিছু বাইরের থেকে তো দেবে না, এরকমই তো আছে।
হুম হুম, ওকে ওকে, ঠিক আছে, তাহলে আমি পরের দিন, হ্যাঁ সিলেবাসের ভেতরেই দেবে, সিলেবাসের বাইরে থেকেও দিতে পারে, কেএনইউ তো ইউ ক্যান সে উইথ কেএনইউ,
হ্যাঁ সেটাও
ইউ ক্যানট সে
সেটাও ঠিক।
উইথ কেএনইউ, সিলেবাসের বাইরে থেকে তো ওইটাই তো প্রবলেম, সিলেবাসের ভেতরে দিলে তো কোনো টেনশন হতো না, মানে সিলেবাসটা না দেখে পুরনো বর্ধমানের সিলেবাস থেকে কোয়েশ্চেন দিয়ে দিতে পারে, প্রচুর এসছে, একবার কি দুবার, কত রাগারাগি হয়েছে আমার সঙ্গে কেএনইউর মধ্যে। এখন আমি বলা ছেড়েও দিয়েছি, বিকজ দে আর বিয়ন্ড অল, মানে কথাবার্তার বাইরে চলে গেছে, কি হচ্ছে আই এম নট বদার্ড, আই এম বদার্ড উইথ ইভেন্টস অফ মাই কলেজ অ্যান্ড মাই ডিপার্টমেন্ট। সো
হুম হুম।
তোমাদের যেসব প্রবলেমস হচ্ছে, আমি তো আমাকে বলো, কারণ আমার পোর্শন শেষ হয়ে গেছে, সো আদার থিংস আমাকে বলো, আমি পড়িয়ে দেবো। ঠিক আছে, মানে তখন
ঠিক
আমাকে
আছে
নোটিশ
স্যার।
বার করতে হবে যে অ্যাজ এ স্পেশাল ক্লাস, কেন আমি তো অন্য কারুর
হ্যাঁ
পোর্শন
হ্যাঁ।
আমি ডাইরেক্টলি আমি পড়াতে পারি না, আই ক্যান টেক আদার পার্সনস পোর্শন অ্যাজ এ স্পেশাল ক্লাস, যে after I
হ্যাঁ
finish
হ্যাঁ
my
হ্যাঁ।
portion, since I have finished your portion I can take other class, others class with a notice called special class or remedial class or revision class or tutorial class, মানে এরকম নাম দিয়ে আমাকে নিতে হবে। ওকে দেন, তাহলে কোয়েশ্চেন যদি না থাকে দেন আমি আজকে এখানে শেষ করছি।
আচ্ছা স্যার।
ওকে ওকে।